Thank you very much. It's an honor to be here. I really admire the students that made all this event. It's extremely professional. I'm very excited to tell you about the quest for personalized medicine and how can our identity detect how exactly we're going to get personalized therapies. So personalized medicine is the ability to offer the right drug to the right patient for the right disease at the right time and with the right dosage, which is very important. So using genetic and metabolic data, we are now able to have drugs to be tailored to specific target patient groups. So basically, over the past few years, the FDA and the European Medicines Agency, they have approved drugs for patients based on their specific genetic profile and based on diagnostic tests. So on 31st of January in 2012, a new therapy for cystic fibrosis was announced. That was a breakthrough because this therapy was tailored for patients that has a specific mutation called G551D. And there's many mutations in cystic fibrosis, which, by the way, is a lethal disease. But only 4% of these patients, they had this specific mutation. And thanks to personalized medicine, the specific patient group was able now to live a healthy life and not have the dramatic consequences of cystic fibrosis. So in personalized medicine, we have a new area for drugs. So before personalized medicine, what used to be done was that one dose and one medicine would fit everyone. So we could give chemotherapy to cancer patients, one chemotherapy for any type of cancer. And that would not be appropriate because some of the patients may respond to the normal dose, which is okay, but some of them may respond to a lower dose. Some of them may respond to higher doses, and some may not respond at all. So using personalized medicine, we can now have effective therapies, we can have the right dose, and we can save millions of lives, as well as the costly health system that is responding to ineffective therapies. But how is it possible that our genetic code, our very own DNA, would dictate the right therapy that we could take? Before we answer that question, let us take a trip to our body. In our body, we have cells, and cells have nuclei. And in these nuclei, we have the chromosomes. I'm sure that everyone knows what a chromosome is. Maybe you don't remember that the chromosomes is basically a line of DNA which is entangled here in this very unique X shape of the DNA, of the chromosome. And if we untangle it, it's about 2 meters of genetic material, so DNA. Parts of this DNA are called genes. So just small parts of DNA will be our genes that we all hear very often these days. And these genes can then be translated into proteins through our machinery in the cells. So proteins are very important machinery in our cells that perform essential functions and help us live a healthy life. So if I ask you what a protein looks like, you'll probably say, this is what a protein looks like, and that's correct, because we have it in our food, we have it in our bodies. We need proteins to survive. But if we take a microscopic look inside the proteins, the food, we will find that these proteins have very unique, specific, three-dimensional shapes that allow them to perform these essential functions in the cells. Unfortunately, we're not the only ones that have proteins. Some proteins are bad because they come with the pathogens that have them. So HIV, RT, influenza virus, hepatitis C is just some examples of pathogens that they depend their survival on specific proteins. And I'm showing some of these proteins here. If we know which are these proteins, we can block them using specific small molecules that are called drugs. We can deactivate them, and then we will have a cure. So drugs block disease proteins. And I'm sure that everyone here knows that drugs are very important. They have actually made our life expectancy 30 years longer since 1950, and they have made us improve our quality of life. But maybe not so many of you know that these molecules, drugs, they're small, organic, very, very small, tiny molecules that have brought this revolution. And I'm sure, I mean, is there anyone here that hasn't taken paracetamol or aspirin, Depon or aspirin? I'm sure everyone has taken, myself included. So it's very, very difficult to create a drug because these drugs have to be very, very specific for the disease that we are targeting. So what happens in pharmaceutical development is first scientists discover the diseased protein. So which one is the protein that the pathogen will base its survival on? If we find the protein that the pathogen needs to survive, if we kill that protein, we kill the pathogen. And that's what we want to do. So we find the diseased protein, and on that protein, we're going to design specific small molecules, drugs, that will deactivate the malicious protein. We need to optimize, of course, the drug because we want to have it not to be toxic. We want it to be metabolized every 8 hours. I'm sure you don't want to wake up every 1 hour and take antibiotics, so that's not very practical, but you also don't want the drug to be in your body for a month. So it needs to be metabolized every 8 hours. So that is called lead optimization, so drug optimization. And only very, very few drugs will go to that stage. They will be tested on animal models because we need to determine whether they have toxicity and efficacy. And if everything goes well, we can go on into clinical phases and that have 3 phases. And after that, if it all goes well again, we will have FDA approval. This is a very, very lengthy process, between 12 and 15 years, and it costs an amazing number of $1 billion. So before having this approach, the traditional way of drug discovery was with random screening of hundreds of thousands of molecules. So basically just having plates with a pathogen and testing randomly different drugs, different chemical compounds. We also had the random discoveries. I'm sure everyone remembers from the high school book how Fleming discovered the first antibiotic, penicillin. Maybe one random discovery that's not so well known is the discovery of Viagra. It's also a random discovery. So initially, Viagra was launched in a clinical trial by Pfizer as an antihypertensive drug. However, in one phase of the clinical trial, it was determined that this drug was not a better hypertensive drug than other drugs on the market for the heart. So it was said that the clinical trial would discontinue, but unfortunately the male patients didn't want to return the drug because it had a side effect, and that was a totally random discovery. Unfortunately, we cannot base ourselves on luck because the estimated number of small molecules that can act as drugs is 10 to the power of 66, so all the possible combinations, but the number of atoms in the world is 10 to the power of 50. So even if we wanted to synthesize all possible combinations of drugs and test it on one specific indication, it wouldn't be possible. That's why we go to targeted therapy approach, which have their base on rational drug discovery. What that is? First, we identify important genes for a disease. What is the protein, what is the gene that the pathogen needs to survive? And we need to deactivate that. So targeting or inactivating these genes or proteins of the pathogens with small molecules, and the concept is actually very simple. It's like the gene, the disease gene, is one lock, and with the key, that is the small drug, we will lock the protein and deactivate it. And that has been possible by using computer simulations, which I'm going to talk about later, in order to visualize the 3-dimensional structure of proteins and design specific small molecules that will be inactivating these genes for targeted therapy. So essentially, what is personalized therapy? It's the quest of getting the population, analyzing the DNA of the population for a specific disease, and then finding the subpopulation for which this specific drug is safe and effective. And it should not be used for other groups where the drug would be safe but not effective, so there's no point. Unsafe and not effective, that's the worst case. And unsafe but effective. So one very prominent example of personalized therapy is for a specific patient group of lung cancer. 4% of lung cancer patients, they have never smoked, and they are below 45 years of age, and still they get this disease. It's very unfortunate, but for them, the scientists found that in their genes, they have a rearrangement of a protein called anaplastic lymphoma kinase, and this, when it's rearranged, it can cause cancer. The scientists took this protein, they visualized it, and on this protein, here with this weird pattern, I'm showing that they managed to create one drug, which is called crizotinib, that is specifically designed to target the mutation of these specific patients. So personalized medicine is not only needed for getting the right drug for the right patient, but also for avoiding adverse drug reactions. So this drug here for treatment of epilepsy and neuropathic pain, there's a risk group of people. If they take it, they have a specific gene variant in Asia, and in Europe it's different. So if they take it, they have a lethal adverse reaction, which is a dangerous skin reaction. So the solution is that before you take the drug, you actually have a genetic test to see whether you have one of these gene variants. And if you have it, you should not take carbazepamine. You should take another drug. So the new drug generation is now based on the fact that we can have diagnostic tests. So in the case of breast cancer, for breast cancer patients, they do a diagnostic test, and if they have a gene that's called HER2 that's overexpressed, then they take Herceptin. So if they have EGFR in lung cancer, they take Avastin. So based on the specific genotype, they would take the appropriate drug that would be effective. Also, we can determine the dose for the people, for the patients, based on their metabolic types, and also whether they would have adverse effects. But how exactly do drugs work? And that's a real fascinating thing for me, and this is what I'm studying. So proteins are polymers of amino acids. So many amino acids go together and form one protein. I'm showing here hemoglobin, which is a protein in our blood. And drugs would actually interact and block the proteins, like I showed you with the key and the lock, by engaging into special chemical interactions. These are interactions is something that we learn already in school. It's the charge interactions, so plus and minus interactions, and other classes like hydrogen bonds and hydrophobic forces. These are all chemical principles that have been applied to the drug that I was showing you before for the lung cancer patients. This was developed based on the interactions that the drug was doing to block the protein. So your name here, so personalized medicine for many indications, that's what the future holds. And a very important aspect of this work is to perform this research more cost efficiently by using supercomputers. And this is the area of research that I'm engaging at the Biomedical Research Foundation of the Academy of Athens, finding cures for medicine and biology using computer power. So what we do is that we compute before we go to the lab and actually spend all this money in expensive experimental equipment. We compute how the protein and the molecule complex will look like in the computer. So there's algorithms that would take big libraries of molecules. The molecule is here. And it will actually tell us, the computer, which of all these drugs or candidate drugs would be appropriate for the diseased protein. So they help us a lot. And here we can watch a video of how this process is working. So like we said, the drug targets diseased proteins. The protein is a three-dimensional structure. This is the gene, the outcome of the gene. And the proteins are made of amino acids, just as they come out of the DNA. translation. So all of the amino acids, they would come in together from the gene expression and form these three-dimensional structures called proteins that would perform these essential functions. But sometimes the proteins are diseased, they are malfunctioning, or they are essential for a pathogen survival. And this is what we want to block. So we have different ways of visualizing proteins, and that's with these serpentines. Let's make it easier for us to understand it. And as we said, these proteins, they have specific cavities, they have holes in them that one small molecule can go and block. So it's like a key locking the protein. So this is an example of one drug. And you can see how small it is compared to the protein. So if you block it at this position, the protein will stop acting and it will stop the survival of the pathogen or of our own diseased cells, as in the case of cancer. And at the heart of designing a new drug is the specific chemical interactions that we talked about, that as chemists can go inside the computer, visualize them, and understand exactly the complementarity of the molecule that we should place in the protein to block it and actually deactivate the protein. And here you can see that the blue molecule, which is a drug, it's actually inside the protein, and it's blocking the site of the reaction of the protein. So here is another example. This is an anti-cancer drug, and you will see how it holds itself together inside the protein in order to deactivate it. So it tightly binds and completely deactivates the protein. So the blue one is the small molecule, it's a drug. And imagine that Depon or Ponstan, it will go into, when you drink it, it will actually go in proteins like that and stay there and alleviate your pain. So this is very, very similar. So basically, the basis of the drug design that we perform is understanding the intermolecular interactions, so the chemical interactions that have to be made in order to design the perfect small molecule that will fit specifically in the diseased protein and will deactivate it. Okay, I think we can go on. And I can also give you more examples of how computers have helped us to understand how drugs work and to design better drugs. So if we can play this first video here. So you see here an anti-cancer drug and how computer simulations have helped us to understand exactly how the protein is, how it moves, and how the drug will actually find the specific location, the specific hole that it needs to block in order to deactivate it. And now let me finish by two case studies that we perform in the Biomedical Research Foundation of the Academy of Athens. I should say that we have a lot of excellent researchers in Greece, and the concept of personalized medicine is also studied in Greece. So this is work from my lab on a mutated protein, which is called PI3 kinase alpha, and it's most commonly mutated kinase in cancer, and especially for 30% of breast cancer patients, they will have one of these two mutations. So in the lab, we studied how is it possible that you have this one change in one amino acid of the 2,000 that PI3 kinase has, and you get cancer. So we used a combination of simulations, so computer work, and experimental work to understand how and whether we can block this diseased protein with a small molecule. So that's another video, if we can play it. So this is a protein, the PI3 kinase. So we simulate, okay, we cannot see it very well here, I don't know what's wrong, but you can see that basically what happens is that we can visualize the motions of the proteins, and we can understand how exactly they move, what kind of locations of binding sites or holes they have on their surface, and how exactly we can block them with small molecules that we can design. So if we take the structure of the proteins, of the diseased proteins, and we identify using the video that I showed before on these specific intrinsic chemical interactions, we can design small molecules that can block the disease. And in this case, we did identify one molecule which we named PI3K10. And based on cancers of mice that didn't have the mutation and mice that did have the mutation that we were working on, we managed to say that the molecule, the small candidate drug that we were giving to the cancer, was having an effect. And the second example is targeting pancreatic cancer, again at the Biomedical Research Foundation of the Academy of Athens. Here it's from another lab, Astradiades lab. And you can see that this is the pancreatic cancer. So this is before the treatment. And after the treatment, this is targeting a specific protein again, which is called MYC, MYC. And this protein is overexpressed in pancreatic cancer. And you can see that for cancers that overexpress this MYC protein, if we treat them with a specific small molecule which is targeting the specific protein, we can get really dramatic results. So, of course, this all is a work of many people. It's not only mine. We do have excellent research in Greece. I should also say that we may be the country with least funding in research, but we are third in Europe in promoting and producing research. So thank you very much for everything.